Welcome, everyone. Good morning, good afternoon, and good evening. Welcome to our Nox Medical webinar, Advancing Sleep Diagnostics, Unveiling the Nox SAS at-home PSG solution. My name is Carlos Teixeira. I'm the Director of Clinical Affairs International at Nox Medical, and I'll be your host and moderator for today's webinar. Now, before we start, let me share some housekeeping rules. First, I would like to start with these two disclaimers that you see in front of you. First disclaimer is that the webinar will be recorded for future reference and distribution. The recording will only capture the panelist audio and video, as well as slides, polls, and/or quizzes. The attendees' participation, such as chat messages, raise hands, or microphone input, will not be included in the recording. Second disclaimer is that the availability of the Nox SAS solution may vary between markets, so please contact your local distributor for further information. We will have a Q&A session and discussions. At the end, we will reserve time for that, but you have available a Q&A session already in the Zoom available for all attendees that you can use to place your questions right from the start. This is the agenda we have prepared for today's meeting. First, Professor Schöbel and Dr. Westhölter will share their opinion. After that, Professor Winck will share his opinion. And then we will reserve time at the end, as said before, for the Q&A and discussions. This is our speakers for today. We have Professor Christoph Schöbel, Professor of Sleep Medicine at the Universal Medical Center of Essen-Ruhrlandklinik, Dr. Dirk Westhölter, Clinical Science at the University Medical Center of also Essen-Ruhrlandklinik, and Professor Dr. João Carlos Winck, Senior Consultant and Full Professor of Respiratory Medicine at Porto University, Head of the Respiratory Unit at Instituto Porto, and also Coordinator of the Sleep and Ventilation Unit at Instituto Porto. Now, I would like to welcome you to the Nox SAS, what we call a sleep revolution. And before even moving forward, I will ask Elisif to help us out and launch our first pool that we have of this webinar. So please, Elisif, if you'd be so kind to help us out launching the first pool. So, do you believe that level 2 at home PSG testing will have a positive impact on the field of sleep medicine? Please do answer yes or no. We will allow a couple of seconds for participants to fill in. We have 50 participations thus far. I would say 95% is saying yes. So this is nice. And this is very good. Okay. I think we're good. So again, 96 already shared, and 96 more. Sorry, not 96, 97%, 98. Okay, so almost all participated right now, or 75%. Okay, and 96 of the ones that participated share that yes, they do believe that level 2 testing will have a positive impact on sleep medicine. So perfect. Now let us start. Again, let me welcome you to what we call the Nox SAS solution and what we call a sleep revolution. And why a sleep revolution? Because we call it the precision of the sleep lab everywhere. So the Nox SAS solution is this new flexible way to record EEG, EOG, ECG, and EMG data during polysomnography sleep studies conducted with the A1S. Now, the Nox SAS solution is just an extension of our Nox A1S with a head cable that you see there and a body cable. Okay, now the head cable is where we're going to, or with what we're going to capture the EEG, EOG, and EMG, and also the Nox SAS body cable where we're going to capture the ECG and also leg EMG. Now, we can see that these are the main differences. All the other signals captured in a normal PSG are exactly the same. respiratory signals, cannula, effort belts, oximetry position activity, etc. So the only thing that really changes is this new and innovative way of capturing the EEG, EOG, ECG, and EMG. Again, the only thing that changes is from your neck up. All the rest is exactly the same. And putting that all together, we will provide you, again, the sleep specialist, with signals that are very, very, very familiar to you. Now, again, precision of the sleep lab everywhere. We call it a revolution for better access, helping you to reduce your waiting list by providing the capability of performing PSG at home, in lab, or even in other locations within the hospital. Also a revolution for better home sleep studies. We know that home sleep apnea tests are not enough for some of the population visiting the sleep lab, and now you have the possibility of, in a simple way, performing at home PSG. And also a revolution for time saving, reducing time for hookup when compared to normal hookups, or even patients in the comfort of their own home without having to come to the hospital for that hookup. Okay? Now, before we move to Professor Schöbel, we're going to launch the second pool. Please, Elisif, help us out. Oh, they were already launched as well. So do you think that level 2 at home PSG would be beneficial for your sleep practice? The vast majority said yes. So this is good as well. And without further ado, please, Professor Schöbel, do share your screen.
Okay, I will try. Hopefully can see it, Carlos.
Yep.
Everything is to be seen?
Yep.
Okay. So thanks for the introduction and good morning from San Diego, attending the this year's ADS Congress. And I'm happy that I have not to present for my own, so that I can share with my dear colleague, Dr. Dirk Westhölter, who will overtake the second part of the talk. Having a look at the actual situation, for instance, in Germany. So I know in every country, it's another way how you near patients with a suspicion of sleep disorder breathing. In Germany, we have a real established guideline by law. That means if you have a patient with suspected sleep disordered breathing, after having had the GP's patient history, the patient will get a portable monitoring which is a 6-channel portable monitoring, which is paid by the health insurance companies. And if there are signs for sleep disordered breathing, the patient is sent to an attended full PSG in the sleep lab to confirm the diagnosis. And having been confirmed the diagnosis, the patient will be initiated with a positive airway pressure treatment, also with fully attended polysomnography in a sleep lab. This is what it's paid by the health insurance companies, preventing also the introduction of new innovative tools, because those new innovative tools, they are not paid and covered by the health insurance companies. The problem is that this will lead to long waiting times, a real diagnostic bottleneck. So the longest waiting time for a fully attended PSG in a sleep lab in Germany is at the moment 24 months. 24 months to be sent to a sleep lab to get the confirmation of the diagnosis and to get the initiation of the first-line treatment, which is positive airway pressure treatment in Germany. So fully attended PSG, so-called level 1, is still the gold standard. But having a look on the private paid sector, so we have a divided sector and divided healthcare system in Germany. There are also the private health insurance companies. Those ones, they are paying the unattended home PSG, so-called level 2, which is done at the patient's home. And honestly, they are very interested to increase the numbers because they can save money. They save the money they would have paid for being at the hospital. They only pay the procedure of polysomnography. We are also running from the Ruhrlandklinik a national funded innovation project. This is paid by the German government. And as you can see here, this is based on the legal pathway I showed you. But with this or within this project, we have the possibility also to integrate new innovative tools. Why? Because the health insurance companies are co-working with us in this project. You see it on the left side. These are legal health insurance companies which are paying for new innovative tools, and they are very interested in having the so-called level 2 unattended polysomnography to be introduced in the German healthcare system. Within the project, we can even send the portable monitoring to the patient's home. And at the moment, we are also negotiating how we can send the home PSG, the unattended PSG, to the patient's home so that the patients can hook up themselves at their homes. But before, honestly, we have to validate whether those new sensors are really comparable to the AASM compliant hookup of the PSG. As you can see here, you see the new sensors on the forehead. They are a little different than the sensors you put according to the AASM guideline on the head to have a good EEG monitoring. But when asking the patients, and this is another focus of our group, what about the patient-related outcomes, experience measures, and also preferences? The majority of the patients, they would decide to have the PSG at their familiar home environment. That means patients want to change the system regarding the unattended polysomnography at their homes. Just for you as an impression, because I do not know whether all of you had the possibility to have a look at the Nox SAS system. Several years ago, we had a clinical research program. This was on a new medication against obstructive sleep apnea. This was a medication of the company of Bayer. Bayer stopped to develop this medication because it has not shown the effects they hoped to see. But within this clinical research program, we had the Nox SAS system in parallel within the same individual for the same night in comparison to the normal AASM compliant hookup. That means we had two different monitoring, PSG monitoring within the same individual. And now the next slides, it's only for you as an impression, as a visual impression, how does the traces look like? We have validated those data preliminary in the German Pneumological Society Congress in 2023, and Dirk Westhölter is at the moment preparing a draft for a publication on these data. Having a look, how does it look like? You see on the upper row the EEG data of the AASM compliant hookup, so the established one, and you see in the lower row this from the same individual, the same epoch, the tracing of the Nox SAS system. Just for your impression, this is the wake state. On the next slide, you see the N1 state with an arousal. On the upper row, the AASM compliant hookup, lower row, the Nox SAS tracing, and you see the same epoch in the same patient of the same night. Here you see the stadium N2, and you can also recognize spindles and K complexes in the upper row, the AASM compliant hookup, and the SAS system in the lower row. This is the N3, so deep sleep, same epoch, same patient, same night, AASM on the upper and Nox SAS on the lower row. And last but not least, you see the REM sleep, you see the eye movements, also in the AASM compliance system and also in the SAS system. And you can detect already here that you will see those eye movements also much more in the EEG channel because you have the sensors on the forehead. Having a look on the current state of science regarding the validation of the Nox SAS system according or against the validated attended PSG system with AASM compliant hookup of the electrodes. This was also done in pediatric population, and you see here a publication on 111 patients. And as you might have had the expression like us, that the EEG signals recorded with the SAS setup had a similar morphology, but consistently lower amplitudes with less power spectra across the analyzed frequency range. That means if implementing the Nox SAS system within your daily clinical life, there could be needed a short introductory training. So we implemented the Nox SAS system right now, several months ago, in our outpatient sleep lab. And at the moment, we are planning also to send the Nox SAS system to the patient's home. But now I will overgive to Dirk to show us other recent publications on this topic. Dirk.
Hello everybody, my name is Dirk Westhölter. So Nox SAS has been implemented in some recent studies. The largest one is the multi-center HIV center from the US. This study aimed to screen for obstructive sleep apnea in patients who have or have not HIV. The interesting thing about that is that the hookup process was explained by normal hospital staff, so no sleep specialists were involved in that. Instructional videos and note cards were provided, and over 960 patients participated. And this study resulted in successful home PSG in 88%, so it's a high amount of successful PSG, although the staff was not specialized. Successful PSG meant in that context all signals were available for at least 3 hours. Good quality was defined as artifact-free data and was achieved in 95% of those successful PSG studies. The reason for unsuccessful Nox SAS PSG was more frequently the oximetry signal than the EEG. So that's maybe also comparable to what happens in inpatient setting. And also a high interscorer reliability was shown for total sleep time and sleep stages. So all in all, the authors concluded that Nox SAS is easy and simple, feasible, and also cost-effective. Although in that study, no real cost-effectiveness analysis has been done. Nox SAS has also been studied in another context, and it has been used in a clinical trial, also multi-center from the US, testing atomoxetine, oxybutynin versus placebo in 62 patients to treat obstructive sleep apnea. And also in this study, Nox SAS montage has been done by the study participants, and they were instructed via video calls. And in that study, there's no negative information that there were technical failure to do so. And in that study, it resulted in less AHI and less REM sleep stage and less total sleep duration in the group that received the treatment versus placebo. So Nox SAS could be also used in that context to show differences in treatment. We conducted a systematic review, our group, on the technical feasibility of home polysomnography. And we concluded, we included 30 studies between 1998 and 2022. And of these studies, we found 5 out of 30 studies were done, the home polysomnography montage was done by the participants, and in 25, it was done by home technicians. So there's only rare evidence on that, and out of those 5 studies with safe applied PSG, 2 of these were with Nox SAS. So overall, we've seen a pooled failure rate of 7.8%, which is quite low. And failures were largely related to signal acquisition. In none of these studies, patient preference was a primary outcome. Nevertheless, it was assessed and analyzed in 11 of these studies, and overall, a preference towards home PSG was shown. So we conclude that there is a relatively low failure rate of home PSG when it's done at home. In home PSG, potentially, and if it's broadly introduced into the healthcare system, will lead to more sleep studies and more data that may need a new infrastructure to handle it and to interpret it and to improve patient care. This year in Germany, we started the Unveiling project, which is a joint project of several research universities and centers of sleep medicine. It's financed by the German government. And the aim is to create a centralized data management that integrates data from the upper part of the picture, from outpatient specialists, and from hospitals in the lower part of the picture, so that throughout the whole patient journey, raw data, polygraphy and polysomnography is available for caregivers and to improve patient treatment, and also to make possible to train AI models for automated scoring, and also to improve phenotyping of patients with sleep disorder briefing. Nox already developed a system for Nox SAS for automatic scoring of sleep stages that has shown this abstract a good accuracy and performance. So that's already available, but with more data might also can be improved maybe.
Okay, thank you, Dirk, for presenting us. Yes, and as you heard by Dirk, we have now the real possibility to have a real integrated patient pathway, perhaps also with a stepped care approach. This is what we are negotiating at the moment with German healthcare insurance companies. So who is the right patient for the right diagnostic approach? Who is the one profiting from a portable monitoring at home for an unattended PSG at home, or even for an attended PSG in a hospital setting. So we are hoping to show this and also to improve the healthcare situation at the moment with those long waiting times as I talked before. Let me conclude our talk. So there are differences in EEG. This is due to the different hookup. You have the forehead sensor of the Nox SAS system in comparison to the ASM compliant placement of the electrodes on the head according to the 1020 EEG system. This might need an introductory training when implementing Nox SAS in your daily clinical routine. It has been shown that there is a successful Nox SAS montage, not only by healthcare professionals with reduced times for hookup, but also by the patients themselves when sending the new system to their homes. We might need future studies to analyze the validity of Nox SAS versus the conventional PSG, as we are doing right now when analyzing the data out of the medication project, having the conventional PSG and the Nox SAS system within the same patient in the same nights. And we have to analyze the role of the Nox SAS system in clinical decision making and regarding patient preferences. But honestly, for sure, this is also depending on the country and the existing healthcare systems. But I think it's worthwhile to discuss because this could even open new potentials. So Nox SAS as a powerful tool is a powerful tool in a digitalized and hopefully patient-centered sleep medicine of the future. And thank you so much. We have come to an end, and I'm really looking forward to discussion at the end of the whole session. Thank you.
Thank you, Professor Schöbel and Dr. Westhölter. Thank you for this, for sharing this, your opinion and great thoughts on the utilization of Nox SAS solution in the research setting, but also the transition from this research to the more clinical day-to-day practice. And that's exactly why we've invited Professor João Carlos Winck to share his opinion on this clinical practice on this day-to-day basis. Please, Professor Winck, the floor is yours.
Thank you, Carlos. I think you can hear me and you can see also my first slide, right? I think my job is much more facilitated by the brilliant talks by Christoph and Dirk. And I think I really going to shorten a little bit my presentation. But first of all, I would like to share, I mean, because I was in some of these phases of sleep medicine, I want to share a little bit how technologies have evolved so far. So a little bit about the history. And you can see what was the sleep lab back in the early 80s. And these brilliant people like Alan Reshape, you know, discovering REM sleep with all these pages of sleep studies of EEG and so forth. So this was the beginning of the history of PSGs. And so after this hospital-based polysomnography, we just started to see the transition to home-based polysomnography based on some of these advantages, right? Like Christoph was saying, so patients prefer to be, to do a sleep study back in their homes. The waiting list will be much shorter because you may have much more available devices to send the patient home, and the majority of the patient just refer that they sleep better in their home, so the sleep quality will be better, and of course less work for the sleep technologists. But of course that these advantages can carry also some disadvantages, because this is not 100% bulletproof. There are some failures, and from some study 4%, others 20%. Some of them, these really high failure rates, that's not very good. The issue is also not video recording. I think that new systems are also allowing this to happen, but the majority does not allow you to do a PSG that uses also the video. If you have a patient with respiratory failure, it's not easy to join together a capnography. And of course, if you need to do a PSG and after to do a MSLT, so a multiple latency sleep test or a multiple wakefulness test, it's not so easy to do. So based on these advantages and advantages, I think that's how the sleep medicine evolved. And the era of home unattended sleep studies were already here for some time ago. And I just showed you here two major population cohorts. So the famous American Sleep Heart Health Study and the SWAN, the study of women's health across the nation. There was two very important sleep cohorts that really used home sleep studies. But of course, this was done with home hookup. So the technologists would have to go to the patient home to hook up, and sometimes again to the patient home to disentangle all the sensors. And so what has happened more recently, and I brought here one of the first studies comparing this other change of paradigm. This is to do unattended home sleep study, but with home hookup versus hospital hookup. And this is what these authors in Belgium have done, showing us that really the hospital hookup was as effective as home hookup in the application of home unattended PSG. So differently from the big studies, cohort populational studies in the US, this Belgium study showed that it was possible just to instruct the patient or even apply the electrodes in the hospital and then send the patient home after, more or less 1 hour of, sensor application. But what is really most exciting, and it's me here in this picture with SAS, is this revolution that Carlos mentioned. So being able to yourself, glue all these sensors at home with minimum instructions and, not very complex instructions, this is really what is the new paradigm that we have nowadays. And as Carlos has shown, and I'm not going into detail, as he has shown very clearly how, and also Christoph, how the Nox SAS setup looks like. And this is the different thing. So this is the new cable for the simplified EEG, AOG, and EMG. And this is the hookup card that sometimes we send it to the patient for him to, remind some of the instructions of the technician, or you can even have a YouTube video that you can also upload and see very clearly how you can do that by yourself. And that's not very difficult. This is one of the pictures that you can see. You can have a mirror in front of you to help you out to put the right electrodes in the right, and this is what you look like in the end. So this is the Nox SAS that has one very important difference from the AASM setup, which is the chin EMG. You don't see chin EMG, so you have, instead of the chin EMG, you have a temporal muscle EMG. And this study, you have seen it already, the feasibility study. So it shows that the SAS, so the self-applied somnography, has a very good reliability. Like Christopher said, you have to have some initial instruction to realize what the signals look like. So the forehead EEG signals recorded with SAS express the lower amplitudes and less absolute power. So you have to understand that. However, the signals express more relative power at very low frequencies in all sleep stages. And this is an example of a comparison of a full hypnogram or a PSG of a Nox SAS AA1S with the standard AASM montage and with the SAS montage on top. And as you can see, difference are really very, very, very slight. And this is what also Dirk has shown you about the major AIDS cohort study that included a lot of patients. If I don't remember, I think more than 1,000 patients. And what comes out from this study that's important for you to realize is that in this multi-center, prospective study, more than 80%, so 84% of the SAS polysomnograph studies were successful. And if we look at the artifact duration, we can see that the artifact duration was very low. So the majority was excellent, very good, and good. And the more than 3 hours of artifact signal were basically the oximetry was the major signal that came out from this study with more artifacts. And so, like Dirk was saying, the reasons for an unsuccessful SAS study in these cohorts was being black, having cigarettes smoking, and cocaine use. So there was no association whatsoever with the educational level, prevalent comorbidity, and BMI. So let's talk about our very short but very self-assuring experience in our lab, a private lab here in the north of Portugal, with a very experienced sleep technologist. The average time that he instructed the patient was about 20 minutes, a little bit more at the beginning. So there were no written instruction, but there was no mobile phone access if the patient wanted. And only one patient contacted us because of some doubts. There was we offered the link with the video woke-up instruction, but curiously, only one patient exceeded that. And the majority of the patients we studied, was the major indication was suspicious of OSA. So we studied so far 22 that we analyzed. We now reached like 30 already, and the numbers are the same so far. So the education level was quite okay. So I was included here, so a PhD guy here, and but the majority was people with bachelor and also secondary school. So the average age was 47. So a little overweight, predominantly male, and as I said, OSA was the main diagnostic in 73%. So we had 100% success with on the first attempt, so we didn't have to repeat any study. The quality of the signals were great, 98%. Good sleep efficiency. So there was only 2 studies with some artifact, but small artifacts, so less than the 3 hours that is considered to have, you know, more impact. And it was the EEG that was the signal that had the more impact. Like Christoph was showing on the left, you can see signals from the Nox SAS. On the right, the conventional SAS, you can tell N3 easily, you can tell the REM sleep, but your eye needs to be trained, as he was saying. So to conclude, I think I've convinced you that this new technology makes a reliable diagnosis in the majority of the disorders. And in our experience, it was a very good effectiveness, very low failure rate. So far, 0%. The patients loved it. There was good user friendliness and very low time with the technician. And I think, like I said before, I think that we need in the future to develop the infrared video, portable video that can allow us to upgrade to a more complete, to assess these video in some cases. So thank you for your attention.
Thank you as well, Professor Winck, for this great insight on the more, I would say, clinical usefulness or your clinical practice. We can now move on for the Q&A and discussion. I've encouraged already the participants to share some questions in the Q&A section, and I'll definitely start. I mean, I think you've both or all the speakers prove, but I would like to clearly reinforce this as a question. In your opinion, is this then feasible to utilize this solution in both the research and clinical setting? I mean, just to open up while I check for the questions. Then, is this comparable to the gold standard? I think also all of the speakers reply to that. And last but not least, in what other settings, that this I think is a very important question, do you think that the Nox SAS solution can be also a useful tool? Professor Schöbel, please, can you start by your own opinion, please?
For sure, yes. As I showed you, it's feasible to introduce this new tool, especially in research. Having a look on the clinical background or the clinical situation, it depends on the reimbursement system for sure. So we introduced this for our private insured patients because the private health insurance company are paying for this. We also introduced the Nox SAS system in our sleep lab. So we have an attended PSG with the Nox SAS system, but honestly, we're saving time with the hookup. So that means we have less time for preparing the patients. This is paid by our health insurance companies because of this. It is an attended PSG, but we are using the new sensor of the attended PSG. But for sure, Carlos, we have the potential to open up the home PSG setting. And for this, I know, especially working in the clinical field, you have to look which is paid by the health insurance companies. And that's why we are negotiating at the moment with some health insurance companies to do this in funded projects or in so-called selective contracts. I'm very curious and also very positive that this will work and that we can also collect data out of clinical routine. but it's feasible and it opens up a real chance to revolutionize sleep medicine, as especially in Germany experienced the last years, because we are also experiencing disadvantages. So not everybody has to come to an attended PSG. We have to be careful in the discussion because it's not a thing that you can decide for a fully attended versus a non-attended, but we have to think over which is the right approach for the right patient. So I think every diagnostic approach has its potential, but in the right patient. And I think this is what we have to discuss also.
Yeah, I see that some of the questions that are being placed on the Q&A are in respect to
Yeah,
reimbursement.
I also saw it. Yeah.
Exactly. So this is one of the major points. Every market, every country has their own specifications. So you need to understand those specifications in order to understand if this is a suitable type of study. But please, Professor Winck, your opinion as well.
Yeah. Well, yeah, like Christoph was saying, I think that for the private office, I think this is a major advance. I think everybody agrees that it's in terms of cost effectiveness is a highly good solution. And also for the research, I think it's amazing. I mean, the possibility of doing, 2-night studies, it's really there. I think that this kind of simple hookup system, it's really making us understand better the sleep across the days and across the months or the years. So there are some very good studies. So the SWAN study that I mentioned, the cohort from the women in the US, was really with the focus of evaluating sleep quality and sleep characteristic across the ages, and that is now getting much more easy with technologies like the SAA, so the SAS, sorry. And like Christoph was saying, we had to know the limits of this system, of course, that patients with severe comorbidities, with respiratory failure, so an attended normal PSG, there are some limits. So like I said, we need to have extra channels for capnography to understand if there is hypoventilation, and that is something that maybe in the future it can also be integrated into this simplified kind of system, right?
Okay, so I have two major questions here. So one from Dr. Diego Garcia Borreguero, asking if this allows to monitor PLMs. And yes, I believe that Professor Winck also shared that one or two of his patients of his cohort had the diagnosis of PLM, and yes, it does allow for leg EMG. Another question from a good friend as well, Mikel Aspiaso. He's saying that a provocative question: do you consider that in 2024, is it still necessary to perform a PSG at home or in lab to therapeutical decisions making for most patients with suspected OSA? This is a very nice question for Jonas as well. Now, Professor Winck, and this one I will start by you and then move to Professor Schöbel as well. Please. Go ahead.
Great, thank you, Carlos. Yeah, definitely. Like you said, in our cohort, we had some patients with associated PLMS, and I can say by experience is a very convenient sensor. EMG from the legs is very good, and the way you apply it, it's very easy. So and it monitors very well like other PSG. So very, very reliable. Concerning in-lab PSG, like I was saying, I think that in the future, and that's my experience, so now I only do a PSG when I have patients with suspicions of nocturnal epilepsy or REM sleep behavior disorder or hypoventilation, where I need extra sensors like oxycapnography or video. If I don't need that, I do, in the other cases, unattended SAS level 2 PS3.
Perfect. Professor Schöbel?
Yeah, I think it's completely right to say and also to discuss who is needing a PSG. And this is what I meant having a look on the patient-centered sleep medicine. So I do not think that we have one solution for everybody, but we have to discuss which is necessary. And I'm completely with a colleague. If you have a patient who has only a sleep disordered breathing and you can have the diagnosis with the portable monitoring, I'm completely with him also to initiate, for instance, a positive airway pressure treatment with a portable monitoring. But I'm also with João. If there is any sign for a comorbid disorder, it's necessary also to have the PSG. And we have also to look on which level we are discussing. Mostly it's discussed also on the reimbursement level. And the problem is if you try to implement new innovative tools, they are not paid by the health insurance companies. So that means even if we are seeing the potential for those new diagnostic tools, if they are not paid, they are not offered by the clinicians so far. And that's why I think we have to develop also the new digital tools and also new diagnostic tools from the state we are at the moment. That does not mean that everybody would need a PSG. It's also my opinion, honestly, but we have to discuss which is the right patient for the right diagnostic approach, and having a look also on new sensors which are arising. You know that the first smartwatches are certified as medical products for screening for sleep disordered breathing. So I think this will dynamically change, and we have to discuss how to implement which diagnostic tool at which point for which patient.
Thank you for sharing that as well. We see that Dr. Westhölter had a baby emergency, so he's in and out. I would love to hear his opinion as well. So we have other questions here from Dr. Dingzu. Are we discussing patients with suspected, only suspected sleep disorder breathing or sleep disorders in general? So please go ahead. Again, back to you, Professor Schöbel, please.
Yeah, so it's regarding every sleep disorder. As João also told us, it's not only sleep disorder breathing. This is the most prevalent form of organic sleep disorders, but you can rule out every other organic disorder by such a system for sure.
Yeah, this is your experience, correct, Professor Winck?
Yeah, the same. So it's not only, you can also diagnose bruxism via your new system. So it's quite convenient because the EMG signal is close to the muscle. So yeah.
Yeah, this is in regards to one of the questions as well. Can it add EMG for assess masseter muscle for sleep bruxism? So we cannot add extra electrodes, but the EMG is being taken by this temporal region, so you'll be able to capture, as Professor Winck is saying. Dr. Dingzu as well, is valid total sleep time more than 3 hours a recommended cutoff for the SAS recording success? No, definitely not. I think there's some mistake in regards to what studies explain of being more than 3 or less than 3 hours of active artifact free. Definitely we still consider, at least our opinion, but again, back to both of you, and now probably Dr. Westhölter that we can hear as well, he's back with us, to the 4 hours at least threshold of recording at least. So right, Dr. Westhölter, we talked a bit on utilizing SAS in other settings. We talked a bit also of what other possibilities. Is it just sleep disorder breathing? We talked in general about when to utilize it or not. So your opinion so far as utilizing SAS as well, please, now that you're back with us.
Yeah, I'm sorry for the small interruption. And I think in that study, the cutoff was 3 hours. I don't know why they defined it as 3 hours and not 4 hours. What was the reason behind it? But in that study, it meant so the cutoff was at 3 hours, meaning like successful performance. I am very a friend of data, and I'm looking forward to have more data. And I think that's the opportunity is to and cloud-based system. So we will need. Can you hear me?
Yep.
Yeah, if Nox SAS will come along with a cloud-based solution, I think it's ready for the future and for future sleep medicine. And I don't know what are your plans with the Nox. Yeah, but I'm looking forward to it, and I'm looking forward to make more database medicine.
Yeah, so regarding Carlos, the recording time. So we will do also a study now in Germany, because we are only paid if we have at least 6 hours of recording.
Okay.
So we will define the successful recording, at least in our healthcare system, having a look on the numbers with at least 6 hours of recording. So this would be for us, honestly, the definition for a successful recording. But I think this is also depending on the healthcare system.
Thank you. Thank you for that.
And I think, if I may add, I think that the criteria for success was at least 3 hours of successful sign-off. So you have to have at least that number of hours of successful sign-off.
That's it, yeah.
Yeah.
Okay. Now, a different question or a different angle. So in the era of AI and automatic scoring, have you tried the automatic scoring of the platform and this specifically designed for the SaaS solution? And what's your opinion? Because there's also a few questions around the automatic algorithm. How many epochs do you need to change or whatnot? We'd also love to hear your first impressions, I would say, about the automatic algorithm. Please. Professor Winck,
Well, as far as I'm concerned, I think our sleep technologists are very, very excited with this. The first impression is that it's really amazing tool. It shortens very, very much their manual, scoring. And we are really seeing very good reliability. That's something that is already been described and is published. So I think that will be absolutely an add-on for these simplified systems. So also the simplified scoring system based on a high reliable technology, which is the AI from your side. Yes.
Yeah, also from my side, so the impression is very good, and it's compliance with the data Dirk presented, and I always let the automatic algorithm run over the data and then rescore several data. And honestly, I do not have so many epochs to rescore as in the conventional PSG system. So honestly, I'm quite satisfied with this. But we will also do research work on this, and also measuring time we would need for scoring such a recording in comparison to the conventional PSG.
Yeah, this is definitely not the topic of today, because I mean the AI would be a webinar on its own.
Yeah.
And also a very good question from Dr. Maria. Is there any exclusion criteria for utilizing the SAS PSG from your side that you would see?
Okay, so for me, it's really a matter of patient preference. We had a couple of patients that were not sufficiently, confident to do that themselves. And like you've seen, even in the AIDS study, there was no difference in the reliability between the education level. That was something that was a little more concern. In our case, we had, pretty high level education people, but some of them were not so high, and the reliability was altogether was very, very equivalent. So I would say that it's basically patient preference. I don't see any contraindications and medical contraindication. I don't know what you think, Christoph.
Yeah, I'm totally with you. So it's not about only patient preferences. I think it's the most important. You always have to look, what about their home environment? Is it the patient who is understanding the right way how to hook up, who's able to get the hook up, perhaps also in assistance with some relatives? So I think this is also what you would have to keep in mind. But I think you can decide for your own. Everybody of you are experienced clinicians, and that's why I think just asking the patient, explaining what to do, and I think you can judge for your own the best who is the right patient for the Nox SAS system. And honestly, the majority of the patients would be.
Yep. I also like to share that, I mean, in the last few weeks, sorry, one of my colleagues also at Nox, we've been traveling a bit around Europe, and we were in one country, that doesn't matter, that the clinicians referred that they were doing not simultaneous, but 2 PSGs at a time in the sleep lab, one in the normal AASM hookup and another one on the SAS hookup. And the patient that was doing the normal, I would say, or standard hookup, look at the other patient and said, well, why can't I have that? That seems way simpler than the one that I'm doing, and seems way more comfortable than the one that I'm doing. So this is really showing that, I mean, nowadays patients clearly are more informed in a way. And if it transmits, or if it, I would say, decreases the patient burden doing such a test, of course, the results we would obtain will be more reliable results, right? Okay, so the time, the hour went down very, very, very fast. Thank you for this. Before we leave, I would love to cover 3 brief topics. So first of all, we can visit our webpage at noxmedical.com/resources, where we have all of these information about SAS and all our other solutions. We will be releasing a Nox SAS clinical compendium in the next few weeks that will gather all the existing evidence around the Nox SAS solution. So yes, please be on the lookout for that. If you're attending Sleep 2024 in Houston, please do visit our booth 1021. We might have a surprise or two prepared for you there. And please do also consider joining us for our social event on Tuesday at the House of Blues. The link's right there. Now, last but not least, I mean, thanking all the speakers for the presence here today, for their amazing perspectives, and thanking you for attending our webinar today.